I think it's a very interesting thing because when you speak in the field, you really hear a lot that notion that cell lines within the same cell subtype, like different pluripotent cell lines, or even other cell lines behave wildly differently. The notion of how differently it is also a notion of expertise, because the more attuned to what you do you are, the more the minute differences seems so big. But when you look at development is very resilient. You basically output almost exactly the same thing, and actually minute difference in development have huge impacts. So that's also the reason why I guess development is so resilient, especially in vertebrates. And so trying to bridge these two notions, knowing full well that the development is a very strong machine, very resilient machine, actually capable also of coaxing behavior of cells. I'm thinking about Ledouarin's experiments showing that cells that come from cancer can be coaxed into normal development by the environment of the embryo. And reconciliating that notion with the huge variability that everyone is speaking about in vitro feels to me like is a testimony to the importance of organizing cells, because it's basically what happens in development. You're organizing cells. The cells are more or less the same, but in vitro, without organization, you get a huge variation, whereas we know that in vivo, development is really, really, really resilient and reproducible.
Yeah, no, absolutely. I totally agree with what you said. I mean, there are so many different variables when we work in vitro, right? So one is the structure. Cells are not organized in any way, right? They are in this very stiff environment, most of the cases, just on top of the plastic, trying to grow. So this is a huge difference compared to in vivo. Then there is the role of the extraembryonic tissue. So we talked about symmetry breaking before. We have many examples of how stem cells can undergo symmetry breaking on their own, right? Without any external interactions. They have this self-organization. But now what we think, based on the evidence from many groups, is that the extraembryonic tissues are there to really make this process of symmetry breaking very robust and always happening in the same place. Whereas if stem cells are doing it on their own, this is more chaotic, sometimes may happen in one place, sometimes in other. So with the extraembryonic tissues, you ensure everything is very, very robust.